Good evening, everyone. Good evening. So I hope I'm audible. Good evening. So my voice is coming to you. Voice vastundi. I am also good. Like, how are you? Namaste. Good afternoon. Good afternoon. Good evening. Oh, doing fine. Nice. So I'm also good. I am very good. I hope you are also doing good. Namaste. How's my day? My day is all good as usual. So I'm trying to make it best. Good afternoon. Yes, I had lunch. What about you? I hope everyone had your lunch. Yes. Shubhyam Madhyanam. Okay, Madhyanam. Good evening means it's a Bagundara. It's a Bagundaru. A few things I know, like how to respond in Telugu, like Bagundaru. You are asking for someone, and your answer is Bagunano if you are good. So anyway, like we have just started our journey. We have started our journey from where? That is a cell, the unit of life, and obviously we are somehow in the midway of the cell, the unit of life. So just we'll try to move forward. So move forward means cell membrane is done. Already we have discussed about the cell membrane. So cell membrane is thin, delicate, lipid, proteins, carbohydrate. Proteins already we have discussed. It's a polypeptide chain, and this protein can be of different types. One is we can say it's embedded, and other one is superficial, or we can say it's a peripheral. So one is integral, other one is peripheral. Integral, peripheral. In integral also fully embedded and partially embedded. So likewise we have started our discussion in the cell membrane. Who proposed? That is a Singer Nicholson, and the name of the model is fluid mosaic model. In this class, we'll try to discuss somehow about the cell wall. Okay. So how many of you are having the cell wall in your body? So I hope your answer will be none, because we are nothing but the animal cells. Animal cells don't have any kind of cell wall. So in the next topic, we'll move to, that is nothing but the cell wall. So cell wall is what? Dead or living? So first structure, or we can say first thing we can add is cell wall is what? It's a dead structure. And if it is a dead structure, means it's a permeable, semi-permeable, or selectively permeable. So for this we are simply writing it's a permeable. It's a permeable. Yeah, Sadhna, it's a permeable. So here it's a permeable, means it's not differentiating between the molecules. It's a not differentiating between the molecules. It allows the molecules to pass through it. Like in the cell membrane, it was selectively permeable. Selectively permeable means only the molecules can enter inside the cells which is required. Other than this, it is not. If it is not required, it won't be allowed to enter inside this. Okay, so it's a dead, permeable, and I hope it should be thick and rigid. So it's a thick and rigid. Can we say that this is universal, means it is present in all cells? It is present in every cells. So obviously my answer is totally incorrect. It is not present in all cells. It is not present in all cells. So we can't say it's a universal. But if you are talking about the present in few cells, and if I am writing the name of the cells, or we can say organism, so the first thing that comes in my mind, so what's that thing? It's a fungus. So fungus is also eukaryotic organisms, and it is also having the cell wall. But that cell wall consists of what? Cellulose-like substances. Cellulose-like substances. Don't say it's cellulose. So this is nothing but the chitin. Chitin, or some pronounce it as a chitin. But don't pronounce it's a chitin, it's a chitin. So chitin is nothing but the component of what? Fungal cell wall. Fungal cell wall. After that, if you are moving to the algae, so if it is written algae, no doubt algae belongs to what? Belongs to the plants. Algae belongs to the plant. But still, we will write here it's a galactans, mannans, and we can say pectin is also there, cellulose is also there. But here we will write galactans, mannans, pectin, protein, cellulose. So many other things you can add here. But the most important part is it's a galactans and mannans. After that, if you are moving to the next one that we are concerned with is it's a plant. So plant is having what? So plant is having the cellulose, hemicellulose, pectin, protein. So these are the different chemical substance. So the introduction, or we can say brief introduction of this chemical substances, we will deal in the one separate chapter is there, and the name of the separate chapter is the biomolecules. So biomolecules is the separate chapters that is dealing with the chemical compositions, or we can say it's a structures. Okay. So anyway, that biomolecules is also interesting chapters. So here we can say plant. In plant, what is a component? So it's a cellulose, and next one is hemicellulose. Another chemical substance that can be present, that can be found is pectins, proteins. So these are the different chemical substance. Yeah, Usha Rani, yeah, yes, calcium in algae, calcium carbonate is also present. Calcium carbonate is also present. But again, the calcium carbonate is not present in all. So there it's one algae that is known as red algae. So red algae is having the calcium carbonate present on its surface. Okay. So after that, what's the doubt, Pranav? What is hemicellulose, Janvi? So cellulose, if you know the cellulose, so cellulose is nothing but the polysaccharides. Same as is hemicellulose is also a polysaccharide. Hemicellulose is also polysaccharides. So that we will discuss in we can say in depth in one other chapter. That chapter is nothing but the biomolecules. So here simply we are talking about the chemical components, or we can say name of this. What are the uses of calcium carbonate? So wherever you find calcium carbonate, so calcium carbonate will provide the rigidity. Calcium carbonate will provide the rigidity. That is the main function of calcium carbonate. Obviously, while making the wall, we are using the cement, we are using the bricks likewise. So it also adds the strength, or we can say rigidity. Strength, or we can say it's a rigidity. Next one is, so this is somehow we can say it's a cell wall. And a brief introduction of cell wall will be like this. So let's say this is cell wall, and cell wall can be of mainly two types, or we can say based on the time frame, it can be chitin is present in saprophytes. We can't directly say that chitin is present in saprophyte, because saprophyte can be of any types. Like fungus can also be saprophytes, bacteria can also be the saprophytes, animals can also be the saprophytes. So that's why we can't directly say that saprophyte is having what? Saprophyte is having the chitin. We can't directly say. But in the fungal cell wall, we can say that fungus is a saprophyte which is having what? Mostly it's a saprophyte, because fungus is also having the parasites, saprophytes, many other nutrition is there. Okay, so directly don't generalize the things because that will give you negative marks. So anyway, if you are talking about the primary wall, so primary wall can be of two types. Sorry, cell wall can be of two types. One is known as primary wall, and other one is known as secondary wall. So one is primary wall, other one is secondary wall, and name itself suggesting that primary wall comes first or comes later. Primary wall comes first or comes later. So my answer is primary wall comes first. Primary wall comes first. That's why we are using the name that is a primary. Primary means which is present in all plant cells. All plant cells. So here I am just writing primary wall. So in this case, primary wall, it is the outermost layer. Outermost layer of what? Outermost layer of plant cells. Outermost layer of plant cells. While in this case, what we can write? So secondary wall, it is present just inside the primary wall. Present just inside the primary wall. So here we can say present just inside the primary, present just inside the primary wall. So this is nothing but the secondary wall. So one is primary wall and one is secondary wall. After that, what else we can add? So the primary wall and secondary wall both contains what? Both contains cellulose. But few special chemical substance which is not present in the primary wall, but present in the secondary wall. What I said, what is the main difference between them, between the primary and secondary is? Primary is having all the molecules, but not all molecules present in the secondary wall is present here. Okay, so a slight difference is there. In this case, we can say primary wall is capability, or we can say is capable to grow. What we can say? It is capable to grow. So next one is here, it is capable to grow. While in this case, we can't say that it is having the proper capability of growth, because almost the modification is done. So there is no further change, or we can say no further growth. Almost growth is absent. Almost growth is absent in the second. Okay. Next one is what else we can add. So other thing is in the primary wall. So primary wall is present in all cells, or we can say primary wall is mainly present in what? Meristematic cells. Primary wall is present in the meristematic cells. And meristematic cells that we will discuss later. Firstly, we'll write present in meristematic cells, or we can say it's nothing but the meristematic tissues. So what is meristematic tissues? The tissues which keep on growing, or we can say which keep on dividing. So any plant cells which keep on, or we can say continuously dividing without any stop, without any pause. If cell division occurs, this type of cells is known as what? This type of cells is known as meristematic cells. Okay, or we can say it's nothing but the meristematic tissues. So present here, but it is, so in the meristematic tissue, this one is not present. It's not present. So here we can say present in what? Present in permanent cells. Present in permanent cells. So one is meristematic cells and one is permanent cells. So meristematic cells means the one which keep on dividing, or we can say continuously dividing, while in the case of permanent cells it just stop, or we can say just pause. It's a cell division, means cell is not dividing further. Cell is not dividing further. After that, what else we can write? So here we can find cellulose, hemicellulose, cellulose, hemicellulose. Again, it's a general thing like pectin, protein. So these components is also present here. Along with some special components. So what is a special components? Here we can say it's a lignin is there. Again, is the lignin is nothing but the substance. It's a substance which gives extra support, or we can say extra strength. So lignin we can say. Suberin you can find. Thereafter you can find the cuticles, or we can say cutin you can find. So a lot more different type of substance you can find where. So you can find in this secondary wall. So the primary wall and secondary wall, the composition is also different. After that, what else we can add? So here we can say, so what else we can add? In this, in the case of it's a primary wall. In the case of primary wall, most of the cells are what? Most of the cells are dead or living. So most of the cells are living here, but in this case, it's a dead as well as living. Both can be possible. Dead as well as living, both can be possible. Okay. So here we can say, while forming the cell wall, or we can say once the primary cell wall is formed, after that which will come? So after that which will come? That is a secondary wall will come. But once the secondary wall is formed, can we say that disappearance of what? Or we can say diminishing of what? Diminishing of primary wall is there. So is there any functional difference between the? Yes, obviously functional difference is also there, but not much of functional difference is there. Because in both the cases, what it is doing? So in both the cases, it is protecting the cells from the outer world. Only the difference is like a structural difference you can find, but functionally, more or less, both are having the same difference, or we can say same function. So here we can say, in this case, most of the cells most of the cells which is having the primary wall is living. While in this case, dead as well as living both can be there. So dead as well as living both we can consider. Next one is what else we can write here. So here we can say in the secondary wall we can write one more thing. This secondary wall what? Diminishes. When it diminishes, when the primary wall diminishes, can anyone tell? So primary wall diminishes whenever the secondary wall is formed, or we can say it is about to the form secondary wall, primary wall diminishes, or we can say primary wall disappear. So here we can say primary wall diminishes. In this case we can say primary wall diminishes as the secondary wall appears, or we can say as the secondary wall appears, okay. After that, what else we can add? So this is somehow related to what? This is somehow related to the cell wall. But after that, two main points that is coming in the need is one is plasmodesmata, and other one is known as middle lamella. So middle lamella and plasmodesmata are the two structures that is somehow related with what? That is somehow related with the cell wall itself, or we can say a plant cell itself. Okay. So here in this case, I am moving one more part, moving to the other part. So this part we can call it as a plasmodesmata, and actually really this plasmodesmata is the most important point. Plasmodesmata. And if the question is coming, plasmodesmata is living connection or dead connection? So what's your answer? Plasmodesmata is living connection or dead connection between the two adjacent plant cells. Usharani, it's a dead connection, living connection. Priya, Russell, living connection. Yes, the answer is living connection. Because the cytoplasm of one plant cells is in contact with the other plant cells. And whenever you are talking about the cytoplasm, means we can say somehow we are talking about the living part, or we can say that is known as living connections. Is that clear? So what is the usage of plasmodesmata? Plasmodesmata means what? It establishes what? Cytoplasmic connection. It establishes cytoplasmic connection between the adjacent cells. Cytoplasmic connection between the adjacent cells. Okay, so it's like you can say that this is one room, and this is the other room. So person from this room can enter into this room with the help of what? With the help of one common door, or we can say one common gate. So that common gate is considered to be as what? Plasmodesmata. Is considered to be as a plasmodesmata. But again, what will happen? Like the cytoplasm from one cell, or we can say cytoplasm from one plant cells enters into other plant cells. But the cytoplasm, or we can say the particle from one plant cells has to cover the thickness of what? Has to cover the thickness of one is cell wall, one is cell wall, and under is nothing but it's a middle lamella. So direct question is coming in the NEET exam. So what's the direct question? What's the direct question? What are the layers traversed by? What are the layers traversed by plasmodesmata? So my answer is cell wall plus middle lamella. So cell wall plus middle lamella. What is mean by chitin? So chitin is again a type of sugar, or we can say it's a polysaccharide. That is the heteropolysaccharides. Is that clear? In this case, what else we can add? That is a plasmodesmata, and this plasmodesmata, it is traversed. It is traversed, or we can say passed. It is traversed, or we can say it's a passed. Traversed through cell wall, and one more structure that will come here is middle lamella. So it has to cross what? Cell wall as well as middle lamella. So these are the two parts which it has to cross. Okay. So next one is, so this is nothing but plasmodesmata. After this plasmodesmata, we'll move to another structure. That another structure is nothing but that another structure is known as what? It's a middle lamella. So other structure, we can call it as a middle lamella. Okay, so in this case, middle lamella, if you are talking about the middle lamella, so have you seen while making the building, or we can say while constructing a building, or we can say making the wall? So in that case, in that wall, what will happen? Like the builders, or we can say whoever is building this wall, or we can say making this wall, in that case, one bricks stack upon another bricks, but in between them, what they are placing? They are placing a cement. So that cement here we can say somehow, or we can say somehow relate with the middle lamella. So can we say that middle lamella is nothing but the plant cement? Middle lamella is a plant cement which holds two plant cells together, or we can say which holds two plant cells together. So whenever you remove what? Remove the we'll remove the middle lamella. Already means two cells. Earlier it was joined tightly. Now it will released. And one more thing is, in the presence of middle lamella, it is getting proper shape. But once you break this middle lamella, what will happen? The cells are loosely packed. Once it is loosely packed, what will happen? So it is not having the proper structure, or we can say definite structure. So in this way also we can say that middle lamella helps to acquire certain type of structure, or we can say certain type of shape. Okay. So can we say that first structure, middle lamella, is nothing but the plant cement? So are you agree with me? Middle lamella, can we call it as a plant cement? Are you agree with me? Because it is also holding two structure together, that's why we can call it as a plant cement. And in the plant cement, what are the materials we are using, or we can say what are the materials plant is using? What are the materials plants are using? So that is known as pectin, or we can say it's a pectate. That is known as pectate. And the pectate is also having some kind of salt, or we can say pectin is a salt. Salt of what? Salt of calcium and magnesium. So you can find the calcium and magnesium from this part. Okay, so here we can say, so it's nothing but the salt of calcium and magnesium pectate. Okay, and next one, yeah, it's like a glue, Rama Reddy. So it's like a glue, or we can say it's a Feviquick. We are frequently using the Feviquick. Lakshmi Devi, what are the function of middle lamella? So the main function of middle lamella is to hold more than one plant cells together. More than one plant cells together. So here we can say it holds tightly. It holds tightly. It holds tightly the plant cell together. Holds tightly the plant cell together. So once you break this, once you remove this, obviously what will happen? So earlier it was packed, or we can say compactly arranged, but now it is nothing but loosely arranged, or we can say some superficial type of structure you can find. Okay. So this is somehow we can say it's a middle lamella. Middle lamella. So in this case, I'm just drawing one structure. Let's say it's a. Let's say this is nothing but the one plant cell. This is one plant cell. And the plant cell is having the outermost boundary is the cell wall. So we can say it's a primary cell wall. Okay. It's not a hexagon actually. Simply I'm just drawing random structure so that everything will come. So this is another plant cell. This is another plant cell. So two plant cells are there. Two plant cells are there. And these two plant cells are connected by what? These two plant cells are connected by plasmodesmata. And one cell wall, or we can say one layer is there. So these two plant cells are connected by what? Connected by plasmodesmata. Connected by plasmodesmata. And next one is, this is connected by plasmodesmata. But at the same time, can't we say that this structure is somehow vulnerable? Somehow it's a weak structure. So if it is a weak structure, so we need to provide some kind of rigidity, or we can say it's a support. Rigidity, or we can say it's a support. So if we are providing the support, so this support is coming from where? This support is coming from middle lamella. So here we can say this part is known as what? This part is known as middle lamella. Okay, so just we'll try to level the diagram. So this is nothing but the middle lamella. This is middle lamella. And next one is what else? So this one is known as plasmodesmata. And plasmodesmata is traversed, or we can say it is passing how many layers? So can we say it's a cell wall? It's a cell wall. And next one is it's a middle lamella. So it is traversed by two layers. One is cell wall and other one is the middle lamella. Okay. After that, what else we can add? Here, if it is required, then we can say inside that, which layer will come? So inside that it's a secondary wall will come. So this is nothing but the secondary wall. So primary wall and secondary wall. So this one is primary wall, and just inside that, what's that? It's a secondary wall, and these are the two plant cells. Let's say it's a mango plant, or we can say it's a mango tree. So here I am just writing. So plant cell A, and other one is plant cell So one is plant cell A and one is plant cell B. Okay. So these are the what is the difference between the plasmodesmata and middle lamella? See, plasmodesmata is a pipeline. Plasmodesmata is a pipeline through which many things can be exchanged. But in the case of middle lamella, so middle lamella is a cement, or we can say it is joining two structure together. It is holding two structure together. Secondary wall and primary wall present in all plants. Secondary wall we can't say present in all plant cells, but even we can say it's a primary wall. So primary wall means each and every wall will start with the primary. But once this secondary wall will form, there is no usage of primary wall, because all the function that was carried out by the primary wall, now it is carrying by what? It is carrying by the secondary wall. Middle lamella is useful in every plant. Oh yeah, obviously it's useful in all plants. Does all plants have middle lamella? Yes, all plants is having middle lamella and plasmodesmata. So actually, without this, no plants can acquire, or we can say maintain the shape of this. Okay, so that's why. What is the advantage of plasmodesmata? So plasmodesmata is having an advantage because it is giving another type of exchange mechanism. Between the two plant cells. If it is not there, we can't say. If it is not there, we can't exchange, or we can say plant cell cannot exchange one thing from this cell to this cell, or we can say vice versa, this cell to this cell. So that is the main function. What about the cell membrane? What about the cell membrane? Cell membrane is also there. So I have not drawn the cell membrane, but cell membrane is there. In this case, now we'll move to the other part, the functions of cell wall. So if you are talking about the function of the cell wall, so what are the different functions of the cell wall? First one means it provides, or we can say it gives a shape of the plant, or we can say plant cell. This is the first part, or we can say it's a rigidity, support, turgidity. These are the different words we can relate to this part. Okay. After that, what else we can add? If it is a plant cell, means outermost layer is cell wall. So how plant cell is interacting with others? How one plant cell is interacting with the other plant cells? With the help of cell membrane, or first structure that will come is the cell wall. So I think the first structure that will come is the cell wall. So it also helps in what? It also helps in cell to cell interaction. Means one plant cell is interacting with the other plant cells with the help of what? With the help of cell wall. So it also helps in it helps in interaction of what? Interaction of cell. It helps in interaction between plant cells. Interaction between plant cell. Okay. After that, what I can add here? It also protect the plant cells from any kind of infection. Like let's say it's a fungus, virus is trying to infect the plant cells. So which structure can protect? So this structure can protect the plant cell. So here also, it also protects from, it protects from infectious agent. It protects from infectious agent. So infectious agent, or we can say any pathogenic substance, or we can say pathogenic thing, we can call it as a infectious substance like bacteria, fungus, or we can say it's a virus. Many other things can be there. So overall, it protects from the infectious agent. Okay. So these are the few things. After that, it is not allowing, or we can say it doesn't allow what? It doesn't allow unwanted macromolecules. It doesn't allow unwanted macromolecules. So these are the few points we can attach to this. Okay. So these are the few functions. Now we'll move to the other part of eukaryotic cells. So can we say that cell membrane is done? Cell wall is done. So once you are done with the cell wall, thereafter it's a cell membrane. So inside the cell membrane, what is there? So can we say that inside the cell membrane, cytoplasm is present? Cytoplasm is present. So cytoplasm means, can we say cytoplasm plus nucleus is also there? So cytoplasm plus nucleus. Yes, good. Sadhana, that is a protoplasm. Good. So inside the cell membrane, what is there? So it's a protoplasm. What is the disadvantage of plasmodesmata? I don't find much of disadvantage of plasmodesmata, because plasmodesmata is advantageous to plant cells. So now we'll move to the another things, that is nothing the protoplasm. And if you are talking about the protoplasm, means we have to consider cytoplasm plus nucleus. And inside the cytoplasm, the first structure, or we can say first thing that we are dealing with is, so it's endo. So endo means inside or outside. Endo means inside or outside. So obviously it's a inside. Endo means it's a inside. After that, one more thing I am writing, endo membrane. So endo membrane, and other part we can add is endomembrane system. So this is known as endomembrane system. So endo means inside, or we can say it's internally. So internally what we are talking? So internally we are talking about a system where internally more than one organelles are connected with each other. Internally, more than one organelles are connected with each other. But again, next question will come. What are the organelles, or we can say what are the type of organelles is internally connected in the system? In this case, my answer is any organelles Any organelles? You told cell wall is selectively permeable, so how? Cell wall is not selectively permeable. Cell wall is permeable. Cell wall is permeable, but at the same time, I am just writing here it's a macromolecules. It is not allowing macromolecules to enter into it. If it is small, it can somehow enters. Okay. So anyway, where we are, so it's endo means inside, or we can say internally. Internally, a lot more organelles are there, which is connected to each other, and that makes what? That makes a system. So one system that consists of more than one organelle, and internally it is connected to each other. Okay. So here I am writing a group of cell organelle, performs coordinated function, coordinated functions, and internally connected to each other. Functionally. Connected to each other functionally. So whoever the cell organelle is qualifying this criteria. So only 4 organelles are qualifying this criteria. Only 4 organelles are qualifying this criteria, which is internally connected to each other. That is not a coordinated function. Coordinated means what? One is cooperating other, or we can say one organelle is cooperating other organelle to perform certain type of function. Okay. So here I'm just writing few group of organelles. What are the group of organelles? So mainly 4 organelles are there. 4 organelles would be there. What are the 4 organelles? So the first organelle, we can call it as a endoplasmic. This is the first organelle. Which is qualifying this criteria. After that, which organelle comes? So after that, one more organelle will come. That is known as Golgi body. Good. Neha, Golgi body. No, mitochondria is not there actually. So endoplasmic reticulum, and next one is Golgi. Next one is Golgi body. Other one is nothing but it's a feature of plant cell, or we can say characteristic feature of plant cell is nothing but the vacuoles, and the last one is suicidal bag. Last one is suicidal bag, that is known as lysosomes. So these are the few organelles which is cooperating each other to perform certain functions. Like a total 4 organelles are there, and how many of them is having single membrane? How many of them is having single layered, or we can say single membrane? Answer me, I'm just waiting for the answer. Out of 4, how many of them is having single membrane? How many of them is having single membrane? I'm just started getting answer 2, so it's a 3. Someone has written it's a 3. 2, but most of the answer I am getting 2. Very rare is 3. One answer I am getting, that is a Muhammad, that is 4. CSK7 also it's a 4. Now I think a trend changed. I'm getting a little bit more number of 4. Yes. So the correct answer is what? Correct answer is 4. Endoplasmic reticulum is having one membrane. Golgi body, again it's a 1. Vacuole, again it's a 1. And lysosome, again it's a 1. So can we say that each and every organelles which is present in this endomembrane system is having what? Is having single membrane. Is having single membrane. Congratulations CSK7, you have given, and one is Mohammed. You have given correct answer. You have given correct answer. That is a total 4 organelles are there, which is having what? Which is having single membrane. So after that, we'll draw the diagram. We'll draw the diagram how it is connected to each other. In this case, firstly we'll draw the diagram of nucleus. So nucleus is having how many membrane? So the nucleus is having 2 membrane. Nucleus is having what? It's a 2 membrane, and these 2 membranes are somehow parallel to each other. But at some point it fuses, it merges, it joins to form what? To form an opening, and this opening is known as what? This opening is known as nuclear pore. This opening is known as nuclear pore. After that you can find one extension of what? Inner membrane or outer membrane. So my answer is outer membrane. So extension of outer membrane. So this is the extension of outer membrane, and this extension of outer membrane will divide what? What's the doubt? Cas compass. What's the doubt? Okay, so just write your doubt, whoever is having doubt. So it's nothing but this is the outer membrane, and outer membrane is having what? Outer membrane is having what? Outer membrane is having extension. After that, this extension will bear what? Will bear small type of granules or like substance. So this actually granules like substance is ribosome. This granules like substance is known as ribosomes. Okay. After that you can find some other part of this, and that other part is having the irregular structure, Okay, so now let's try to level the diagram. So in the leveling of diagram, firstly we'll level this. It's nothing but the nucleus, and after this nucleus, this is surrounded by the nuclear membrane, and this is outer membrane of nucleus. This is nothing but the outer membrane of nucleus. Next one is inner membrane of nucleus. Next one is here it's nothing but the ribosome, but what type of ribosome? ATS type of ribosome. ATS type of ribosome. And if this part is having ribosome, so this part we can call it as a rough endoplasmic reticulum, while the other part which is lack of, or we can say where is there is absence of what? Absence of ribosomes. This part is known as what? That is a smooth endoplasmic reticulum. And the space which comes, the space of cytoplasm which comes under this membranous thing, this is known as what? Luminal. This is known as what type of space? This space is known as luminal space. And outside this we can call it as a extraluminal. So outside space is known as, so here we can say it's nothing but the extraluminal space. Okay. So this is nothing but the diagram of what? Diagram of endoplasmic reticulum. So can we say that endoplasmic reticulum is extension of what? Extension of the nuclear membrane. Outer membrane or inner membrane? It is extension of outer membrane of nucleus or inner membrane of nucleus. So it's outer membrane of nucleus that will give you what? Endoplasmic reticulum. And endoplasmic reticulum can be of how many types? So it's a 2 types. And so first type which is having the ribosome, other type which is lack of ribosome. So if it is having ribosomes, means it will synthesize what? It will synthesize proteins. If it is having ribosome, means it will synthesize protein. Means RER will synthesize what? Protein. And SER will synthesize what? Other than the protein. Means we can say it's a fat, lipid, or we can say lipid related substance like sterone, or we can say testosterone, or we can say many other sex hormones are there that is also prepared, or we can say synthesized by what? Synthesized by SER. So RER and SER is having the same function or different function? So I think it's a different function. One synthesizing protein, other is synthesizing fat and lipid. Okay. So these are the few differences that is there between what? Between the RER and SER. Okay. So if you are going to gym, or we can say doing some kind of exercise, so which one is good for you? RER or SER? So if you are doing gym, so which one is good? RER or SER for you? So I think RER is little bit good because it will synthesize what? Protein, and here it's nothing with the muscles, or we can say it's a protein is there. Okay, but at the same time, muscle movement is also there. So whenever you are moving your muscles, or we can say muscle contraction is there, it is controlled by SER. It is controlled by SER. So while doing gym, what will happen? SER is more activated, but after the gym, RER is required. RER is required. Okay. So likewise, these are the few things that we have discussed in the class. So for now, it's done. We'll meet in the class if it is there. So For now, it's a bye-bye to everyone. Take care.